group that's here, I think this is really a doubly exciting meeting for lots of reasons, but you've got the chance here to talk to one of the leaders in this whole field of genomics, and he's going to talk about that. But it's a rare opportunity. I have an opportunity from time to time to meet with or talk with leadership that do the same kinds of things John does. But I think Dr. John Mathews, is interesting because he's the senior clinical development leader at 23andMe. And that means that he is the clinical lead for all of the non-oncology programs and supports all of the therapeutics, providing clinical direction and leadership for drug development and programs to discover novel genetic targets and deliver novel medicines. Here we're focusing on, obviously, IPF. So Dr. Mathews, may I say, it is a pleasure to have you this morning. And we're going to change our view a little bit here. And I know you have some slides to share, so if you bring those up, and we can start anytime you're ready.
Great. Well, thank you very much for that introduction. Very kind of you. Welcome, everybody. It's a great pleasure to join the PF Warriors and to have a chance to start this conversation with you. I think of this as the start of a conversation. I hope not to be technical or to bamboozle or to confuse anyone. So if we need to come back and do a redo, please reach out to Bill and let's not leave anyone behind. So I do have slides to share. I'm going to take through an introduction, let you know a bit about myself. We're going to question, why is 23andMe studying IPF? We've come together today because you all have either got this condition or know somebody or care for somebody with this condition. So we are very much focused on you and IPF. Bill's asked me to define and to give a background as to what is genetics, so that but this will be very high level and not scientific or confusing. I'm going to introduce you to our research platform, what we do at 23andMe, and share some insights from an IPF study. This is research of 23andMe research participants who've consented to share their information, and we've summarized it and shared it back to your community, and I want to take you through that data. I'm going to share some descriptions of privacy and data security. And I'm also going to describe an ongoing study which we started a few months ago, an IPF research study which is ongoing. And this is an informational presentation, so it's not about recruitment, but really about information, so that Bill can make sure that you're all aware of this study. And I'll lastly touch on how the IPF study data are being used. So Bill gave a very kind introduction to me. Yes, I'm a senior clinical development leader. I've been just under 2 years at 23andMe. I'm a physician. I trained in the United Kingdom. I have a PhD in respiratory clinical pharmacology from Imperial College in London. And I trained, and I had years of experience in general medicine and specifically pulmonary lung medicine at the Royal Brompton Hospital in London and at the Lane Fox unit at St. Thomas's Hospital in London. Before 23andMe, I had 10 years at Genentech. I was involved in the development of a number of molecules for mainly focusing on lung disease. And I did help design and conduct a trial in interstitial lung fibrosis with the molecule lebrikizumab. So on 23andMe, I work on drug development programs to discover novel genetic targets to deliver new medicines that we hope will be provide better medicines, better efficacy, better safety, good therapeutic window for the treatment of interstitial lung fibrosis and other diseases. So why are we studying IPF? You're obviously very familiar with your condition. This is your community. But I wanted to share my high-level comments so that you realize we're on the same page, and hopefully we can have a common understanding of IPF. So I understand it's a chronic and progressive lung disease. Misdiagnosis is an issue with this disease, and we can talk about that in the discussion. And the length of time to diagnosis is definitely a problem, and this is an area that we need to work on. There are approximately 250,000 affected by IPF in the US. So it is what we call a rare disease, but it's not so rare. It does affect a large number of people in the United States and around the world. The course of the disease is highly variable. Some people progress fast and need lung transplantation, and there is a mortality associated. So it is a very serious and severe condition. There are 2 FDA approved treatments for IPF, aspirin and nintedanib, but there is need for more treatments. We're sure of that. The existing treatments have issues with tolerability. Some people have to stop taking them. There are ways to manage that, but it's not always perfect. And we know that you can discontinue the treatment because of these tolerability issues. So there's definitely room for us to improve. So our long-term goal is to develop more effective treatments with fewer side effects that can improve the lives of those living with IPF to help them live longer and better lives. And we believe at 23andMe, to get there, we need to learn more about the genetics of people living with IPF. So I just want to be very clear about our mission. We're helping people access, understand, and benefit from the human genome. So what Bill asked me to do is well, what is this human genome? It seems kind of scientific and a bit confusing. On the left there is a bold statement, which I believe in, and I think it's a good statement to ground ourselves in, that unlocking this genetic code creates the opportunity to revolutionize the diagnosis, prevention, and treatment of most, if not all, human disease. So that's a great, bold statement, and that needs to be our mission statement. So cracking the code, what is it? These ACGTs, these are letters that are just names of the specific elements, the building blocks of DNA. I'm sure you might have heard of the double helix, the structure that was so excitingly discovered, and this chain of instruction that sits in each of our cells in our body that is a bit like a recipe. The cell takes the instructions inside the nucleus, the center of the cell, and every cell follows these instructions to make specific proteins. But it's a data problem. It's a very big data problem, and 23andMe has sort of set out to find a way to use the code in this way that we can develop better medicines. So we're 99.5% genetically alike. It's a surprising statement, but when we actually look at our DNA, there's about 6 feet of it all coiled up in each cell. And when you look at the code, we're 99.5% genetically alike. And there's 3 billion base pairs, so there's an awful lot of information there. So when I'm talking about genomics and genetics, what we do with 23andMe is take a saliva sample that allows us to extract DNA in sufficient quantity that we can run it on a chip, and I'll show you that chip. It's not sequencing, but it's what we call genotyping. And so we're able to look at specific places along the entire genome and compare somebody with IPF to somebody that doesn't have IPF. So I'm going to introduce you to this research platform. And I want to start with a series of statements that is fundamental to the 23andMe approach. Firstly, participation is easy because it's online and it's decentralized. The kits can come to you, and it's remote and online. Geography, therefore, is not a barrier. So everyone can do it from their own environment. And we can include everybody in multiple studies. IPF patients will not only help us understand IPF, but actually through all the information and the health of every individual, we actually learn about an awful lot of diseases. And so it's inclusive. There's what we call a research feedback loop. We make discoveries, We share discoveries with the participant, with the individual, and we hopefully create educated and engaged participants that can then help us make more discoveries. So there's what we call a research feedback loop. I do want to share that I think it's more complicated than that simple 3-point circle. And PF Warriors is in here. If we talk about research participant, or we talk about communities, I think we should specifically add it to the biotech, pharma, and academia that there are communities that represent rare diseases like IPF. So there's patient advocacy. In the middle here, we have 23andMe research, and we're feeding the data that comes from our participants that consent to participate. And it comes to me here at Therapeutics with the team, and we can then develop drugs and feedback to research participants. We do have part of the 23andMe reports are FDA approved, so our health reports around specific risks for diseases are FDA approved. So there's a participant-driven ecosystem here to understand genetic basis of disease. So the platform is about participation, understanding an individual's genetics, and then grouping it together. The surveys, this is individual questions, patient, what we call self-report. Somebody will be asked, do you have IPF? And they will be able to answer yes or no. And that's part of our survey data that allows us to make these discoveries. So where does it start? Step one is collecting consent. So it's opting in. It's a opt-in process. It's IRB approved. That means that an institutional review board reviews our protocol and our consent forms, and as I would say, gives us permission to start. So it's all overseen by this research process of having what we call ethics approval. We have broad protocol and consents. So we ask people to consent for research broadly. So it wouldn't be a specific, let's say, IPF consent, but to participate broadly. Customers can cease participation at any time. So that means you can stop and you can opt out when you want. And the data is aggregated and anonymized. So the anonymization means that you as an individual, when it comes up to the research, we're not looking at you at an individual at that point. We can't link you back to your personal address or your specific information that we aggregate. That means we group it all together and we look at group data. So although an individual has participated, it becomes part of a group in terms of understanding. So this is the step two, is once we've collected the saliva sample, this allows us to get enough cells sitting in the saliva that allows us to extract DNA from the cells. And we use what's called an Illumina bead chip. So on the right is a photograph. For size, we're talking about, I tried to look up the exact size of it. I have actually held one of these. You can hold it in your hand. It's about, say, 4 inches by 1 inch. It's an Illumina, what we call an Illumina chip. Illumina is the name of the company that produces them, and we customize them for 23andMe with our specific elements inside the chip. And what it does is it looks at the genome at hundreds and thousands of genetic variants. So although all those base pairs are incredibly long and lots of them, we look at specific points along that so that it can all fit onto one chip. And we use a process of imputation. This is it's a statistical process that allows us to effectively guess the pieces in the middle that we don't measure. So this is called genotyping. It's not sequencing, so we don't measure everyone's individual exact DNA code along every base pair. We do what's called genotyping, where we look at these on this Illumina chip. There are videos on the 23andMe database. We can share more information. This process has been put onto videos, so you can see what happens in the lab if you're interested and want to know more details. And I'm happy to come back to this. I know it's a bit technical, but I wanted to share kind of what is the brick and mortar behind our process. So this is about step 3, which is collecting the phenotype data. So your communities, I'm sorry, I've got you on the bottom row. That's only because these are stacked in terms of size. That doesn't mean you're bottom of priority. But the idiopathic pulmonary fibrosis group today, as of today, we have 4,300 participants that have consented for research. And so we collect from those participants. They've told us they have IPF. And what I'll show you in a few slides' time is how we've aggregated that data and how it actually does reflect your community. And so we're very excited that we're actually collecting we're collecting people with IPF. We are looking at another rare disease called systemic sclerosis, and we also have a specific ongoing study for that disease as well. We collect patients who tell us they have sarcoidosis. We have 6,200. So this is to show that we do look at all diseases, and if anyone comes to us with a specific disease, they'll fit into their own bucket, and we'll group all together the people. And obviously the more common diseases, diabetes and high cholesterol at the top, that collects a lot of people. But you can imagine you could have both IPF and high cholesterol, and you'll be in both places, and you'll contribute to the discovery of drugs in both places. So the analysis. So I started by trying to describe the genome, and so in the middle here, if you just cast your eye to these two double helixes side by side, this is to try and represent two individuals. I think we can call one with a smiley face and one with not a smiley face. So the yellow and the orange, and the light blue and the dark blue are representing the bases in a little piece of DNA, just a little visual representation of a couple of turns of DNA. And what we can do is we can compare through that chip that I showed you, what are the specific A, T, C, or Gs at each side. They pair because they have complementarity. They join together in pairs, and we can measure them at specific places on that bead chip. And what it allows us to do is to see what is the difference for a John with IPF or Bill with IPF versus somebody that doesn't have IPF, and we can show the difference, whether you're an A at this place or a T, whether you're a C or a G. And that's called a GWAS. It's a genome-wide significance test to show us the confidence that we understand the difference at those places. And that ultimately will lead us to information that says, well, what's happening? Because these DNA strands lead to proteins. They lead to actually how the cell operates. And that's where we need drugs that can interact with these proteins and either switch them off or make them more active. So to give you a sense of scale, if you just look at the graph on the left-hand side, the x-axis there, 2015, going year by year up to 2020. And this blue line is the number of what we call independent hits. These are places where we've shown a statistically significant difference. So that's where we can see that the difference between that person with the disease versus the person without, there's enough of a difference that we think that it wasn't just a random chance finding, and that if we did the study again, we would see that same difference. And so it's repeatable. And so it's really secure and interesting data. And what it's saying is between 2015 up to 2020, we're now up to 40,000 independent hits. And so this shows the growth of the database comes when we get more people coming to contribute data. Another way to show this, if you look at the panel on the right-hand side, 2016, 2017, and 2019, this is for a disease, osteoarthritis. You may be familiar with the joint pain of osteoarthritis. It's quite common, and it can particularly affect the joints, hip, knees, the thumb. And so people tell us whether they have osteoarthritis or not. And when we look in 2016, you can see a few of these reddish lines that are looking at the top. These are called Manhattan plots. You can imagine New York or Chicago, the sky rises. As you look along, the x-axis is the chromosomes, so the place on each of the chromosomes that make up the total DNA on the bottom. And you see in 2016 that there's a red line there at the beginning on chromosome 2. But then in 2017, it started to get joined by lots of other red lines. And in 2019, looks like we've got a whole new skyscrapers have come up. And so we've got a whole bunch of new information that tells us more about osteoarthritis. And the same will happen with IPF as we collect more people, bring more information telling us that they have IPF. The same process happens. We see this across all the diseases, that this increase in what we call significant important hits just increases by the number of participants. So these are really important slides. It does look a bit technical and scientific, but hopefully I've made it accessible, and I'm happy to come back to them. So what about specifically IPF? So I told you that we had 4,300 people tell us that they have IPF that are consented for research. And so this actually turns out to be one of the largest genetic studies of this condition to date. And it's certainly comparable to what we call public GWAS. These are academic researchers, the doctors that look after you when you visit them in the clinic. A lot of IPF doctors are involved in research, and they've also done some GWAS studies. And so what we can do is we can have discovery in 23andMe, and we can do replication with what's public and shared. And we also hope to ultimately we will share our data as well, and it will become public, and we'll present it in scientific journals. 23andMe has at least 150 publications and is certainly committed to sharing data. So what have we found in our study? I'll show you some pictures in a moment, but just to start with, we found that people with IPF tell us that they have gastrointestinal disturbance, the reflux that you often have as a problem in IPF. And so that tells me as a doctor that the participation in our study reflects the IPF that I see as a doctor when I see people in the clinic. IPF patients in our study told us they have sleep disturbance. And so that's an important finding. Shortness of breath, clearly it affects the fibrosis in the lungs, makes someone with IPF short of breath. And so to see that in our report of the symptoms, that's important, and it tells us that we're collecting the right data. We also found an increase in reported migraine and allergy symptoms. I think this is interesting data. We're looking into what it means, but we summarize all of the data, and everything that we're told gets aggregated. And if it's important and statistically significant and clinically meaningful, then it gets shared. So we have over 2 million responses to our survey, including questions about IPF and pulmonary fibrosis. So we're collecting a lot of data. So let me show you a bit more about the insights from this IPF study that we've shared in a blog post that's been published, and it is accessible, and we can make sure you can get the address if you want to go and find it. On the left-hand side is a blue graph, a series of blue bars that show different age groups from 19 through to 99. And each bar is a 10-year gap, a 10-year period. And the y-axis, the height of the bar, shows the prevalence, the number of people with IPF in our database. So everyone that comes into our questionnaire. We ask, how old are you? And we're able to put everyone in this group data. And it shows that IPF, as we see in the clinic and as you experience, increases with age. So the prevalence of IPF, the number of people affected, increases with age. And we observed the highest prevalence amongst individuals of European descent. This was followed by African Americans, and then individuals of East Asian descent had the lowest prevalence. And so this looks at, this is important so that we understand who has IPF, and we can look at the difference in the genetics between these populations. On the right-hand side is the geography of IPF in our customers in the United States. And what we've done here is we've color-coded it, so it's shaded by numbers of people. And what we see is that the observed geographic patterns reflected the effects of age and occupational exposure. So states with an older population like Arizona and Florida have more cases of IPF. And so that's understandable in that it's tracking where these exposures are. And states with mining, such as West Virginia and New Mexico, had a higher prevalence also of IPF, which is consistent with the miners that through occupational exposure end up with an increased risk of IPF. So this is group data from the 4,300 people that we already have. And what I'm going to go on to do is to describe for informational purposes that we've started a study to invite 1,000 people to participate, so we can get some new cases, so we can go from 4,300 to 5,300 and really try and get more information so we can get a better understanding, so we can start to develop some new medications. But I'm going to pause there and just go on to data privacy, because obviously this is a very important topic when we're talking about our DNA. Our customers consent to participate and actively opt in. It's not an opt-out process. You have to actively participate and want to come into this process having read the consent and read all of our disclosures telling you how we look after your data. There's independent third-party institutional review board oversights to the ethics oversight and approval of all of our studies, all of our research protocol and consent processes, and customers can end participation at any time. Within the enrollment flow, participants in the 23andMe IPF research study consent to share their data in multiple ways. They consent to the 23andMe research. This allows 23andMe to use the genetic information and the self-reported information like, do you have gastrointestinal reflux or do you have migraine? That allows us to use that information, and it allows us the sharing of aggregated and de-identified data. So we take an individual and we aggregate it with all the other information, and we also de-identify it, so you can't go back to that individual from the group data. We consent to sample storage, so we consent if the consent is given, then we store the sample for additional genetic analysis as they become useful. And we have consent specifically to the IPF research study, which allows us to de-identify this individual data with a third-party researcher for pulmonary research. So we do actively build into our study that we will ultimately share this data with the other pulmonary doctors in the world who are studying this disease. So I'm just going to end here on this specific ongoing IPF research study. It's kind of a part of the overall, the 4,300 that we already have in a study. We've got this additional 1,000 patient effort. So we are enrolling 1,000 participants with diagnosed IPF or Hermansky-Pudlak syndrome. So Hermansky-Pudlak syndrome is a very rare, much rarer than IPF, but it has commonalities with IPF, because within this condition, the lungs also get IPF. The histology, when we look under the microscope, the lung fibrosis and the disease in Hermansky-Pudlak syndrome is very similar to IPF. And I also included it because I am aware that the Mansky-Pudlak syndrome community were very involved in some of the early studies of pirfenidone. And so I thought it was very important that we include this group in our study. At the moment, I think they're only representing about 1%, so it's a much smaller group, but they're part of the IPF community, and so we wanted to include them. And we're partnering with the PF Warriors to directly reach people with IPF. This is our conversation today. This is informational, and we want to share what we're doing with you as a pulmonary fibrosis warriors community, so that you're aware of what we're doing. So we would like to enroll 1,000 participants who have been diagnosed with IPF or Mansky-Pudlak syndrome. The study is performed under the supervision of a research ethics committee called the Institutional Review Board to ensure concordance with federal regulations and ethical standards. Eligible participants are asked to enroll on a landing page that we provide. That goes through the enrollment criteria. There's a consent form, and this allows us with the sharing of the aggregated, the individual and aggregated data with qualified third-party partners. An individual would then take our baseline survey, complete a short longitudinal survey, so we reach back out to an individual at 3, 6, and 9 months to get an idea of progression. And also a participant would provide a saliva sample for the DNA testing using what we call the 23andMe Health and Ancestry Service. So this is the kit that would be sent out, and this would be provided at no cost to the participant. So the process is one of inform. That's where we're at today. Share information with advocacy organizations and online. We'll then move to an enroll phase where participants enroll in the specific IPF research study. They would take a survey and receive a 23andMe kit. We would collect data, we would process the samples, and then we would do the ongoing 3, 6, and 9 months ongoing collection for those participants. And then we would analyze the data. We use the data for our drug discovery efforts. And lastly, we will share the information, and we will share the insights of our study and make them available not only to the scientific community, but to the advocacy community as well. So I'm going to stop there. Thank you. And questions. Bill, I'll hand it back to you, and hopefully we'll have a good discussion, and hopefully if not confused everybody. Can't hear you, Bill. I think you're on mute.
Bill, you're muted.
I'm sorry. I apologize. Hey, what can I say? I first and foremost, Dr. Mathews, your presentation was truly brilliant. It cut through all of the noise and got down to the meat of what we're interested in. What is the study and how can it help us? I think some of the questions that are outstanding, which I'd like to ask our members to ask and be open about it, to help you understand after this is all done, I'll be sending out an email to you with instructions on how to get more information at 23andMe, how to sign up for the trial and be part of it and what has to happen. And that'll happen automatically, so you don't have to kind of worry about taking notes and as we talk. But this is such an important topic. It really is, I think, the future of medicine. And it's about more than just PF. That's our focus. I think it's going to change the face of the way medicine works and the way treatments work in the future. So it's so important. And you have an opportunity here to have one of the true leaders in that whole field. You look at genomics, and Dr. Mathews is right up there at the top of the pack. So it's an opportunity to ask questions. Don't be embarrassed about them. It's just us talking. And I know some have already dropped me a notes about this and that, and I haven't answered them because I wanted to save them. I know some of you will be looking at the technology per se, some about the results, the expectations, what it means. So with that, I'm going to try to hold my breath and keep quiet unless you ask some questions. I already got a question. It says, John, will the kit also give the participants information on their ancestry that might be considered findings?
Yes. So as an individual, you would be part of the ancestry kit part of it, and so yes, you would see that part. I'm a customer, and I can see one of my first cousins joined, and so I was notified that I had a DNA relative, and so I was able to connect with my cousin Anthony. So yes, they as part of a 23andMe customer, you would get to see your ancestry.
Dr. Matthew, I have a question. I participated in the, I guess must have been the initial test, provided samples like in late summer, early fall. Is this just an extension of that survey, or is this a entirely new survey?
So that the 1,000 study that I described, that started, sounds like it started after you participated in 23andMe. So yes, the study is for the 1,000 people is new, and that involves reaching out specifically at 3, 6, and 9 months. Any customers prior to that time that we started that study, sounds like you're part of the 4,300 if you consented for research.
Right.
We follow up every year. So there's an annual follow-up for health profile that asks people to tell us if anything new has happened with them in the intervening time that they'd like to share.
So when you're talking about share, does this mean that a researcher can contact you and ask you for certain data with qualifications for his research, and you will provide that?
Ultimately, aggregated data is for the 1,000 patient study, and individual data could be shared with a qualified researcher. Yes, it takes time for the whole study to finish, so we'd have to wait for the completion of the study. There's a period of time that's needed for the analysis phase, but ultimately, when it's ready, yes, a third-party researcher could contact us and say they have a specific question, and that the consent for the 1,000 group would allow us to share that back to that specific researcher.
I'm getting questions left and right, and I'm asking people, please ask Dr. Mathews some of those questions. I don't even pretend to be a doctor.
This is Nancy Lee. I'd like to ask a couple of questions. I'm a regular 23andMe customer, and I do have HPS. I've been in the database for several years now, and I must have answered something about pulmonary fibrosis because I do get periodic surveys about IPF, but I've seen Hermansky-Pudlak as well. So I'm not certain if I should really be answering IPF questions because HPS, pulmonary fibrosis, is technically not IPF. Can you guide me on that, as well as do I participate in this 1,000-person survey in addition to what I'm already doing?
If you're already a 23andMe customer, then you've kind of already participated. So we're looking for new people that have not already in the system. So Nancy, it looks like you've already participated, and your data would be used to study what you tell us. So if you tell us you have Hermansky-Pudlak syndrome, and you tell us you have lung fibrosis associated with that condition, we would analyze it in that way. And we do recognize the difference between Hermansky-Pudlak syndrome and IPF, but we wanted to be inclusive for the reasons I described. So we're looking for new customers. We're looking for new people with IPF so that we can increase the numbers of individuals and get the scale that we need to start to make some meaningful discoveries.
Okay, thanks for that clarification. And just another quick question. Have you reached out to the Hermansky-Pudlak syndrome network for partnership in this study?
One of our genetic counselors is very familiar with that group and is in touch with them. So yes.
Okay, good, because I'm on the board of that organization, so I'd like to help if I can.
Great, thank you.
Nancy, knowing you're a loyal PF warrior, we would hope that they come to the PF warriors as well.
Yes, I do spread the word about our PF warriors too.
Dr.
we'll continue.
Mathews, sorry, Dr. Mathews, can I ask you a question?
Sure.
Have you, probably not related, but have you heard of a drug called N115 put out by Emphy Corp, EMPHY-CORP? And they've actually gone through 3 clinical trials, but I don't know how far it's got.
I've not heard of that one.
Okay.
Let me take it down and I'll get back to Bill. I'll do some.
Okay.
Can you say the name again?
It's N115 for the clinical trial, and it's Emphy, EMPHY Corp, CORP is the company.
Yeah.
And it just sounds like it would be a game changer if things.
Steve, if I may comment, there's there's currently about 30 trials in different states and phases around the world on PF, pulmonary fibrosis, and ILD, and that's one of 30. So it's hard to name them all in this kind of a meeting. We're here to really talk about 23andMe, but I'll try to get some answers for you because I think it's an important question. And perhaps we can get somebody to speak to us about all the trials in process so we get some understanding of kind of what's around the corner. I think it's an important question.
Okay, thank you.
I had a question about your sample. Are you looking, or might you look in the future, at familial PF? So for baseline, are you asking for family history of pulmonary fibrosis?
Yeah, we do look at family history and ask people if they have a family member that's affected. So that's one of the ways to look at the genetics, is to include in the analysis people that have the condition or people that tell us that they have a first-degree relative with the condition.
Okay, because I've managed 4 kits. I bought kits way back when you were, in fact, doing the health information before the FDA stopped you on that. And I have my father's sister who had her father, her brother, and her nephew, all of whom had IPF, but she's apparently not, got it, which we're grateful for. So you mentioned the HP, the Hermansky-Pudlak. Are you also looking at other biomarkers like telomere-related disorders like TERT? So dyskeratosis congenita patients can often develop pulmonary fibrosis and sometimes have a TERT mutation other than a dyskeratosis congenita mutation. So I don't know if that's a group you've thought about at all. Smaller numbers, rarer than PF, so it's a small sample.
Sure. So the rare disease that you describe, that anyone that purchased a kit themselves and became accustomed and told them that they had any rare disease would be included in our rare disease analyses. And so if there are, I think, more than 5 individuals, we can start to do some numbers, because if it's less than 5, it's hard to be aggregated. So yes, we do have a rare disease look across the 7,000 rare diseases. In terms of that specific telomere protein that you're looking at, yes, because of the genetics of that, we would look, that would be included in the GWAS analysis. And if that came up as a significant protein, we would be able to look at that one. But to be clear, we don't measure in the sample, we don't measure telomere length. We don't do any biomarker at the moment.
Right, and you're relying on self-report, right? You're not in a position where you can verify diagnoses. You're not looking for CAT scans. I mean, obviously, you're not in a position where you can look for verification from docs or CAT scans. You're relying on self-report, I presume.
It is self-report, but I can ask you, have you had a high-resolution CT scan, or have you had any CT scan? Self-report is interesting. We did a question that we published on, are you a morning person? So I'm actually not a morning person, but if you ask this group, are they a morning person? If you just think about it yourself as an individual, I bet about half of you will tell me that you're a morning person, and about half will say that you're not. And if we look at a GWAS, so the genetics of that question, it gives us a lot of significant hits, these GWAS hits for what we call clock genes, genes that are involved in how cells and the body respond to light, what we call the circadian rhythm, the response of the body between day and night. And it's fascinating. And the genes that we see, we published on this, gives us the clock genes, and those have been tested in preclinical models, and we have some good links to human genes. So self-report is an interesting way to go, and we believe that it's an interesting way to have this research process that I described, where the individual is able to participate, and we're not having to go to all of your doctors and say, hey, tell us about, and can we collaborate about your patients? We're coming directly to you.
Right. And I don't want to take more time, just two more quick questions. The only thing I've noticed from looking at Inspire is people get confused and sometimes think they have IPF when they're told they have fibrosis, but they might have another form of ILD, hypersensitivity pneumonitis or whatever, NSIP, and then they read about IPF and they freak out. So you could have somebody responding that they have IPF when in fact they have a different form of ILD. The ILD may still be of great interest to you, but so two more quick questions. Go ahead. I'm sorry.
No, well, just to respond to that, no, I recognize that there's an issue with self-report, and the doctors that we've described this study to, that was one of the first things they said was, well, many of the people that come to us with, say, IPF, we decide that they don't have IPF, but they have these other things that you've mentioned in your list, like hypersensitivity pneumonitis. That's another common condition that we have to distinguish in the clinic. What 23andMe have shown, I think, through our scale and our process is that we can get insights, and we may have some, what I would call some error within each point. But when you look at the scale that we have, we're able to get these insights just because of the way it seems to be working, and that you can, I would say, tolerate a degree of imprecision if you get enough people to come on board where there is some accuracy at some level.
Sure.
Dr. Mathews, on that point, if I can just interrupt, I just wonder, is 23andMe ever going to get to the point where you submit a spit sample and you find out, through your analysis that you thought you had IPF, your doctors told you had IPF, but in fact, your genes don't match the pool that you've collected over, hopefully years at that point? Will you think 23andMe will ever return to the patient and say, maybe you should talk to your doctor because you don't match this gene pool?
I would say that we're at the beginning of this process. The reports that we feed back to customers for health reports have to be FDA approved. So for a condition like IPF at the moment, if we were saying that you had increased risk of IPF, we would have to have a report that met the FDA standards. I think what you're describing is definitely an interesting ultimate goal would be to have sort of individual health checks on the basis of your individual genetics. We are doing it for things like health and wellness, where we can feed back reports based upon your risk. So we have some reports actually in it's within subscribers for non-alcoholic fatty liver disease, where we can show based on your individual genetics what your risk is based upon what we call polygenic risk scores. So I don't think we're quite where you're describing, but so I'll leave it at that we need to have FDA oversight and approval for specific health reports.
Do you think an organization like LiveWello will get to that point? Because right now, as you know, you could send in a spit sample to 23andMe, get your analysis back, and feed that into LiveWello, and that will show you all your genes and all your polymorphisms that you could share with the doctor. Like, can that be a loop for this group?
I would say as a physician that's definitely you can get your raw data back as a 23andMe customer, and if that helps you, I can't vouch for. I'm not familiar with the name of the group that you mentioned, and I can't vouch for how the quality and the understanding of all of that data would be, when it's passed on. So our mission is to empower you as an individual, and yes, if you want to use your data in that way, then hopefully it will help you. But I can't vouch for it because it would be beyond 23andMe.
Dr. Mathews, one of the patients had asked a question in text, and Regina, I wonder if you might ask Dr. Mathews your question, Regina Bulliard.
Yeah, I was wondering at what point will you be able to share any of the information that you've obtained through the study with participants? Are you waiting for a certain number of people to be able to get that information? How many more participants do you really need to get a significant amount of data?
I think we need the full 1,000 in order to complete this specific study, in order to be able to share that data and to be able to write a similar blog that we've done, and a description of the data from the 4,300. So I think it is around collecting complete groups, tranches of data, such that the data could be meaningful.
I wonder if Heather, if you could ask your question on transplants, please.
Hi, I am a post-transplant patient and for 10 years, but pulmonary fibrosis is in my family. So I was just curious, if someone like me, can I participate, or is it just current patients pre-transplant?
No, I don't. I think you could participate. I think that, there's a specific enrollment process. I wouldn't exclude a transplant patient because I don't believe a lung transplant would affect the DNA that we collect. There are certain circumstances where, say, if you had a bone marrow transplant, that does change the DNA and would be problematic. But I don't believe a lung transplant would be a problem.
Okay, thanks.
Dr. Mathews.
Hello.
Yeah, I got a question. I'm a Latino. Are you have plans to reach out to the Latino community in Spanish? I know the U.S. is the second largest Latino community in the world after Mexico.
Hello.
So I just have that question. If there's
Oscar, it's a great question.
Dr. Mathews, may I address that because it's so important? Oscar, we have launched a new Spanish language initiative as part of PF Warriors. We will be doing Spanish language meetings. We will be doing Spanish language literature. We are launched, we are live online, and I can't pronounce the name because unfortunately, even though I launched it, I don't speak Spanish. But we have excellent people that do. In fact, Nancy Lee, who is on this call, is part of our steering committee on that group. Nancy, could you just address Oscar for a moment and tell him what we're doing?
Yes, hi, Oscar.
I'm going to ask you to do it in Spanish for me because I can't.
Okay. Oscar, do you want it in Spanish?
No, go ahead in English so everybody can hear it.
Okay. We have launched a Facebook page. We've just started, so we we're inviting members to join us, fibrosis, o guerreros de fibrosis pulmonar. And so you can find us. It's a public page, and you can join us. And that's where we're starting. And we are going to, as soon as we get the number of members that we feel will substantiate a substantial call, we will start our Zoom calls with our Hispanic members. So we're really excited about that, and we're looking forward to growing that group as soon as we can, so we can get all of those things going.
So you say we can find that Hispanic outreach.
Yes.
Where is it? On Facebook?
It's on Facebook, and it's called Guerreros de Fibrosis Pulmonar.
Okay, gracias. Thank you.
Okay, de nada, de nada.
Nancy, you say that so well, I wish I could.
We'll work on that, Bill.
Yeah, so I'm sorry, Dr. Mathews, but I just had to interject on that because it's such an important initiative, because if you look at the data distribution demographics on patient communities around the world as well as the United States, there's this bump for the Hispanic community. And in the United States, in Texas in particular, the Spanish community, the Hispanic community, is a dominant community numerically. And so we're addressing that community, not just here in Dallas or in Texas or in the United States, but we have members in Spain, we have members in Peru, we have doctors that have joined us from South America. So we're very aggressive on it, Oscar. And I would hope when we get done with this call, you run over to Facebook and look up that site and join us, please.
Thank you. I will. Thank you very much.
Thank you, Dr. Mathews. I'm sorry.
Dr. Mathews, I'd like to ask where your IRB approval comes from. Is it an academic institution? And if so, are you partnering with them?
We have an IRB institution that resides in California, and it's independent to 23andMe. We don't have a specific academic collaboration for this study at the moment. PF Warriors is our advocacy group that we do have a specific effort with leading to today.
Thank you.
I did reach out to the Pulmonary Fibrosis
Foundation.
the foundation and the IPF Foundation. So the two large advocacy groups that are aware of our study, and we have we've got some follow-up meetings with the PFF.
But we are number two, Linda, but we do try harder.
You're not number two.
You're number one, but
Do you have, though related to that, do you have a medical advisory board or consultants who are researchers at some of the other medical institutions, even though you don't have a formal association with those institutions per se?
So when we reached out to pulmonary fibrosis, the PFF, I spoke with Gerd Cosgrove. I have spoken to him.
Yeah.
Hmm? He's
Who has
just
left
left.
the
Yeah, I
foundation,
know.
right.
I know. So we'll keep in touch. We've certainly. I've been involved with investigators over the years. So we'll definitely keep in touch. We don't have any specific contracts with any academic institution at the moment.
I meant more individually, like did you consult with Naftali Kaminsky or someone from Vanderbilt or someone from Colorado? Or
if I can comment, because I think a lot of that is very germane and might be difficult for Dr. Mathews to comment on, but I can tell you, I personally am involved with Dr. Kaminsky, know him well, we talk frequently. Giselle Jenkins, I talk frequently with in the UK. I can go down the list in the US, but I can tell you that many of the top tier researchers are involved, and foundations. Kristen at the IPF Foundation is an active supporter of the PF Warriors in this initiative, and I go on down the whole list of other foundations that are. There's an enormous amount of interest here because this is the future of treatment for our disease, and it's coming and it's moving with rapid speed, and I personally am so excited about this relationship. I know you are too, and I know our members are, but kind of back on track, I wonder if some of you have questions about, here's an opportunity. I feel like an auctioneer gone. Here we are with one of the world's foremost authorities in this area who lives it, sleeps it, and breathes it. And we're not taking the time to ask them the good questions we'll ask each other later on. And here's your opportunity, so go for it.
Dr. Russ Amling from Tucson, Arizona. I noticed in your slides, Arizona was one of the most recognized. I have pulmonary fibrosis along with valley fever on top of that. And I've also had polio. Have you had anybody in your studies that have had some of these other, such as you made a list of a few, but with infantile paralysis and this type of thing, do you have anybody that's listed that way? Or would you be interested?
I would certainly be interested. I haven't looked at valley fever specifically, and post-polio syndrome. I used to look after patients with post-polio syndrome at the Lane Fox unit when I was a junior doctor. So I'm aware of those issues. I'll go back and look at the database and see if anyone's told us they have post-polio syndrome. But that sort of combination that you have as an individual, I mean, I haven't looked at it specifically because it's unique to you, but in the principle of looking at combinations of diseases is there.
Dr. Mathews, you've been talking a little bit about some self-reporting, and you gave a list of some related things, including GERD and migraines. And, you know, there's always been this long-standing discussion of, does GERD actually contribute to IPF? But when you talk about migraines, is there any way for you to distinguish, even in the self-reporting phase, between someone who has true migraines and someone who's really just hypoxic often and ends up with bad headaches?
That's a great question. I think you know, you're coming up with a hypothesis as to why migraine is linked to IPF, and I think that's where our efforts are hypothesis driven. I think what we try and do when we talk about what are better questions to get at understanding a hypothesis, I think we would have to ask more questions and go back to individuals and say, you know, tell us more about your migraine. Do you have like aura? Do you have visual changes with your migraine? But to be honest, I don't know. I mean, as a hypoxic patient with IPF who gets headache, I'm guessing that individual won't have the aura that's associated with migraine. So perhaps there's a way in self-report to distinguish those. It's a very interesting way you're approaching it. So I'll certainly think about that.
Dr. Mathews.
Hello.
Hi, it's Jerry Banford here. And my question is, being that I'm probably one of the few Canadians who are a member of this IPF group, are you accepting IPF patients from Canada in your study?
Fortunately, we're not. At the moment, it is limited to the US. I think this is to do with IRB oversight and just limiting the shipping and the location. I think it'd be great if we could expand to Canada, but at the moment, my understanding is we're not.
And if I may comment, Jerry, I just had 3 days of meetings with Sharon Lee of the Canadian Pulmonary Fibrosis Foundation in Toronto. And we have actually at this point in time 33 patient members in Canada, and we will be doing events in concert with the Canadian CPFF over this coming year. And she's just a great leadership, I think, for you. And we have many of the members in Canada that contribute on a regular basis. So don't feel like the Lone Ranger. You're one of us, and we're one of you.
Thank you.
And 23andMe is available in Canada. I mean that's one way to participate. I appreciate it's becoming a customer.
Okay, when it is, that I think is what you said, is that right? When it is available in Canada, or it isn't it?
No, it's not at the moment for the research study, but I'm saying at 23andMe itself, you can join from Canada.
I see. Thank you.
Excuse me, Bill and Dr. Mathews. I'm in Canada also, and I would absolutely be a good study because I have at least 8 in my family that have IPF, and possibly 2 more. Anyway, just saying.
No, it'd be great to have the research study expand out to Canada, and I hear you're affected and your family's affected.
Sheila, we might, would you follow up with me afterwards, please? I'd like to introduce you to Dr. Brown in Colorado, who's doing a study at National Jewish and University of Colorado on familial pulmonary fibrosis. And you are somebody he will want to talk to.
Okay, good. That would be great. Thank you.
Is this an awkward silence? Do I have to start calling on people?
Dr. Mathews,
sitting there, Victoria. I can see you.
I have a question. We talked about GERD, and I just recently saw something that people who with IPF also have hernias, hiatal hernias, along with the GERD. Are you finding this?
To answer your question directly, no. The self-report, the data that we've looked at as using gastroesophageal reflux as a question, as a physician underlying it, it could be the hiatus hernia that you're describing, could be the cause of that. But we're summarizing that the symptom, the self-report that somebody says that they have reflux problems.
There is some things that have been said about Gerd causing IPF. Do you know anything about that?
Yes, I think the underlying hypothesis is that somehow with the reflux of the acid that's meant to be contained in the stomach is somehow aspirated, perhaps in an unobserved way. People are unaware that it's happening perhaps during the night, certain times during the day that's just unaware, and that's an insult to the lung, that the lung responds with fibrosis. So I think there is a hypothesis around why gastroesophageal reflux disease might be implicated in IPF. So yes, that's something that I think doctors have talked about for many years.
Dr. Mathews, as you were designing this, and even as you were considering taking that position at 23andMe, what is something you wish you could do with either that study or your position that so far you've been unable to do? Is there something about this that somehow this group could help influence?
I think what I've, we struggle with it is just, we mentioned 7,000 rare diseases today, and we're starting with IPF and systemic sclerosis as 2 of them, where we've got specific 1,000 cohorts that we're trying to do specific studies for. So I think what we're trying to do is to get the message out that, we'd like to study all rare diseases. And you can imagine that's just a huge problem of scale. How do we get out to each group like yourselves? And we understand all of the issues and concerns that have been raised today, privacy concerns, and the question is how can we advance medical understanding with this research network, this research community that I described, where an individual participates and then we make discoveries and feed that back. So that to me is just seems some days an overwhelming challenge, is how can you get out to enough people and how can you really make a difference for each of those rare diseases?
Dr. Mathews, the two people that I haven't heard from that I'm surprised are Patrick Fernandez and Therese Genovese. I know you're both on the call. I don't want to embarrass you by showing your video. Can you ask your questions, please?
Can you hear me, Bill?
Perfectly.
Okay. I think, boy, you put me on the spot. All right. I think this testing is great. For those who have hypersensitive pneumonitis and IPF and all that, it really doesn't matter. We got to survive and live a good quality of life. However, there's an aspect that we don't know. One of the biggest problems for survival is rejection. Is there any genetic or biomarkers or DNA testing that will tell us how many of us would get rejection from a new lung? It also depends on the immune system. The other part of that question is, will there be regions in the US and in the world that are more prone to pulmonary fibrosis and specifically to IPF? This question on GERD, whether it causes IPF, for me, definitely. Because I think I got IPF from my stomach, not necessarily acid in the esophagus, but gases coming from the stomach that gets into my lung. Because recently I ran some tests and it worked. It proves that out. So biomarkers, and then for people who have had lung transplant. For the people who have not been diagnosed yet, the entire family should have DNA testing. So they know to recognize the symptoms. It takes 3 to 4 years for us to get diagnosed. It took me 4 years. So that will be tremendously helpful for the entire family structure to get tested if they have a history of diagnosis. That's the other part of it. That's all I have to say, Bill. And I don't know how to turn on my camera. It's not turning on.
No worries. No worries.
Yeah.
I mean, I think in terms of your question about lung rejection, obviously transplantation is that end result for very severely scarred and fibrosed lungs. And so transplantation is a hope and a medical solution but we know that it's challenging to give everyone that needs a transplant. And as you highlight, rejection is a problem with lung transplantation. We're not looking at that. We're trying to get to that much earlier phase where you talked about in terms of trying to get to earlier diagnosis and interventions that can prevent the need for having a lung transplantation at all. You mentioned regions around the world. I showed you the plot by state in the USA. I think we have a start to try and look at differences in prevalence across areas. But certainly, as Oscar pointed out, we're not in different languages around the world. We're mainly around English-speaking countries where the burden of translation and so on. But yeah, there's plenty more scale that this could expand to. So hopefully, I've touched on your points. Thank you for bringing them up.
Thank you, Dr. Mathews. We've hit our time. And I have to say thank you, and I just can't tell you how much I feel that is a genuine thank you for what you've done here and talking to us. And thank 23andMe for their generosity and their support of the PF Warriors and their involvement. And this is going to be a great test. I will be sending out an email to all of our members and a link to go to on 23andMe for more information and the opportunity to participate in their clinical trial based on your desire and need and also qualifications. So with that, Dr. Mathews, I want to say thank you. Thanks to our group that's been here, our warriors. You're always there with good questions and good insights, and I'm always personally just amazed how many smart members we really have. And I want to say thank you to our membership, thank you, Dr. Mathews, and thank you, 23andMe.
Thank you. Appreciate the time today. All the best, all of you. Thank you.